Just before lunch, Dr. Miller, you were going to explain or elaborate on parvovirus. Could you take a couple of moments to do that right now? And in regard to that, may we have, please, Exhibit 1513, which is also Canada's tab 10, page 12.
Can we move to page 11 first? Oh, okay, sorry, it was page 10. Okay, move back to 12, sorry. I'm sorry about that.
Do you want to have
the one on the contrast between the retroviruses and...
Oh, 13.
Oh, it was 13, I'm sorry.
Page 13.
Okay, I know I was asked about retroviruses and what retroviruses are, and in certainly one of the stages in my hypotheses about this MRS signature was that it could be elicited from a retroviral-like infection. We didn't end up identifying a retrovirus in association with that signature, but we did identify a parvovirus. And I just wanted to point out that there are some very interesting similarities between the two, despite the fact that one is a DNA virus and another is an RNA virus. And one of which is parvoviruses can insert their genetic material into the host genome. This is not something that is as well known. We have done the work to establish that this is not the case for the sequence that we have. If they've inserted their genome into the host, every cell would have the same complement of DNA, and we would see this virus in every cell within an individual, and we do not see that. Interestingly, one of the most powerful aspects of the signatures in terms of a stress response is a host DNA damage response. And both of these viral families require the elicitation of a DNA damage response in order to complete replication. And so that's something they have in common. They both are associated with very strong immunosuppression of the host. They both actually can cause leukemia-like disease. Retroviruses obviously contain the leukemia viruses, which we know to be causative of leukemia, but there is a plethora of studies over the last 20 years about the role of parvovirus B19 in humans in leukemia-like disease and its role in exacerbating the outcome for people with leukemia. But there are some papers out that show that you can actually get a leukemia-like response from a parvovirus, which is something we also found in our data. Both have been linked with cancer, but while retroviruses can cause cancer, parvoviruses are actually anti-cancer activities. So they target rapidly dividing cells to facilitate their reproduction, and so they've been used. One kind of human one has been used to fight brain tumors, actually. And they can both be involved in latent infections, and both viral families are known for remaining inactive for years to wait until the right conditions to begin to replicate and cause disease. If you can just go to the previous slide, I won't spend a lot of time on this. But in terms of parvoviruses, some of the best known parvoviruses are parvoviruses in dogs, and they can have decimating effect on puppies and young dogs. And there's a parvovirus that is quite virulent in humans called B19. It's associated with fifth disease in children, but it requires hematopoietic cells to divide, and it's associated with severe anemia. Again, anemia is something that comes up over and over again in some of the observations that we have in salmon, not we personally, but people have been observing anemia in salmon, and parvoviruses can actually cause anemia in a variety of different organisms. I think one of the most interesting things about this family of viruses is not only that they require rapidly dividing cells to facilitate their own reproduction, but they can be oftentimes stimulated by stress. And there's a number of studies that have shown that that one can induce, if you have cells that are not rapidly dividing, one can induce the proliferation of parvoviruses by simply stressing the cells. And so I think this is interesting in the context of whether or not a virus like this could become more active and elicit more disease in a situation where salmon are known to be highly stressed when they go between freshwater and saltwater and return back into the freshwater environment. That, at this point, I would say is speculation, but it is something that I'm quite interested in terms of this particular viral family.
Okay, thank you. Dr. Garver, before lunch, you gave some evidence about the lab studies that you have done, and then moving on, lab studies that you're currently embarking upon. Laboratory studies are in a controlled setting, of course. Can you clarify what next steps you see beyond those laboratory studies that you're about to take on right now?
Yeah, sure. I guess to back up just a little bit, I'll give you some thinking, rationale for our thinking on why we're progressing to laboratory studies. This is not a typical disease investigation. Typically, when we approach a disease investigation, it's usually because we actually have a disease or a pathology, or even more specifically, mortality associated in a population. So if there's mortality, if there's a die-off event in fish, we'll go out and then run a disease diagnostic on those fish to identify the etiological agent that might be responsible. And to do that, that involves traditional methods of culturing the pathogens and doing histology to identify the pathology associated. And that's important because once you have the histological marker or signature of that disease, then you can actually see what damage is being done and get an idea of the impact of that pathogen and consequently the disease on the population. And so in this case, it's quite a bit of a reverse scenario. And we first Dr. Miller first identified a genomic signature that might be linked to a negative impact on the fish. And so inferring upon that genomic signature, it was found that there could be a possible virus associated with it. Now, granted, there was no mortality associated with that or a disease associated with it. It's now a sequence of a virus, and so we have to identify, is that an infectious agent, and does it cause disease? And so to do that, you then take it back into the laboratory and do the transmission study. So that's really the key on where we're going. But it's nice to take it into a laboratory study because then you can obviously control the setting, control the parameters, and it has to be repeated. It has to be at least confirmed that you can do that over and over again. And so in that regard, once you establish the fact that it is infectious, it's all under the context that it's in the laboratory, and then once you have histological markers, then you can go out in the wild and start looking at it using histological markers, using the molecular techniques that Dr. Miller is establishing, getting viral loads in these, and really assessing, is there an impact due to the infection of this pathogen on a population?
All right. And my final question then is of you, Dr. Miller, and Dr. Garver has just spoken to some of this, but a lot of this. Do you have anything to add to what's been said there about the approach you're embarking on and doing versus the traditional approach to studying a disease?
Yeah, first of all, I would correct one thing that Kyle said, that it's correct that we didn't have observable mass mortality events, and we don't have observable mass mortality events in the ocean, and we do have mortality in fish when they come back in the river, but we don't generally observe that mortality unless they're dying at the spawning grounds. And when we originally identified this signature, it was associated with mortality in that initial study that's in the science paper. But no, I think it's an important point, and you just have heard two days of talks or of a panel of disease experts who have basically suggested that it is nearly impossible to study a disease in wild salmon because we can't observe their mortality. And if we use the classical approaches of only studying disease where we can observe massive mortality events and assuming that we're going to pick up all diseases by only looking at massive mortality events in hatcheries in one environment, in the freshwater environment where we can see these sorts of things, I think it's clear, given the lack of data that exist in terms of diseases in sockeye salmon, that approach simply hasn't worked, and the approach that we're taking is is a different approach to, and again, we didn't set out to look for disease, but it is almost backwards of what a normal approach to studying disease would be. In normal, microarrays are actually used in the human medical world all the time. They're used to study disease and host response to a wide variety of pathogens and as well as drug therapies. And that's the last thing one does in order to figure out what's the best prophylactic treatment, what's the best vaccine treatment, how do we get an effective response so that animals are less prone to disease from specific pathogens. You know, we flipped that on the head and said, let's just look at what the animals are telling us. Let's look at what the physiology of the animals can tell us about what's affecting them, and then we'll go back and hypothesize as to what could be causing that. And then we'll go back and do the laboratory studies to validate those hypotheses and to validate whether or not one can, at any stage in the development of salmon, elicit disease and mortality associated with what we now have as a candidate virus.
All right. Thank you. Thank you, Dr. Garver, Dr. Miller. Those are my questions, Mr. Commissioner.
Thank you, Mr. Commissioner. Next, I have counsel for the province at 55 minutes.
Mr. Commissioner, Callan, C-A-L-L-A-N, initials T-E, appearing on behalf of Her Majesty the Queen and Ray, the Province of British Columbia. Dr. Miller, how was the common genomic profile defined, and specifically the MRS?
I'm not sure what your question is.
Specifically, is it a specific cluster of genes?
Oh, okay, I'm sorry, yes. The signature, actually, when we first uncovered the signature, we had simply run a t-test between 12 fish sampled in the marine environment that made it the spawning grounds and 12 fish that went missing. That was the very first time we uncovered that signature. However, we added more fish to our study. We did a study in the freshwater environment as well. And when we added those additional fish, a simple t-test didn't pull it out very well. One of the reasons for that, and something that has to be recognized, is that in return migrating salmon, there is not likely a single cause of all mortality that occurs in the river. And so we recognize at the very outset that it may be difficult to simply assume that all fish that die in the river die of the same thing, and that really wasn't our assumption. We were looking for genomic signatures that were associated with poor performance, not necessarily causative of all mortality in the river. We found that we were able to identify this signature with principal component analysis, and we used principal component analysis as a method to identify the major physiological trajectories in the data. And then we looked at correlations between success and lack of success in terms of migration along each of the principal components in principal component analysis. I know that's very technical, but you asked.
And I get what I asked. I understand from your earlier evidence this morning that you stated that the power of the test strength of the signature alone might be evidence of disease. You would agree, however, that the signature alone will not cause disease, although it may be a marker.
The signature alone will not cause disease in and of itself. The signature indicates a lot of activities at a cellular level when salmon reach the freshwater environment. The signature does not cause the disease. Whatever's causing the signature could cause disease.
Now, I understand earlier you were talking about principled and unprincipled. Is that the same as supervised and unsupervised analysis?
No. Principal component analysis is an unsupervised analysis. So it's a way to simply let the data speak for itself and tell you what are the major trajectories in the data. A supervised analysis is if we were to run a t-test or an ANOVA and say, I want to contrast what genes are being turned on and off in this set of individuals compared to this other set of individuals. So that is one of the analyses that we did try, which was to simply run a t-test comparing fish that made it to the spawning grounds and fish that didn't. But in order for a t-test to be to resolve anything very powerfully, you have to have, it depends on your sample sizes, but you have to basically have, you know, a single signature that's associated with whatever you're looking for.
So to summarize it, in your ocean tagging study, the supervised analysis was not statistically significant.
In our first analysis of the ocean tagging study, the first fish we ran, we did actually get a significant t-test. However, when we added more fish to that analysis, because we were looking actually at muscle tissue and gill tissue at the time, and we found that we didn't really get any signal associated with survivorship in muscle tissue. But when we did our first analysis of the gill tissue, we actually did. When we added more samples, it became more obscure. But we did then pull it out with PCA analysis.
Now, in the principal component or the unsupervised analysis, you did find gene expression patterns. And this was the basis for your statement in Exhibit 558, your paper, that 60% of the fish contained a gene expression signature in seawater greater than 200 kilometers from the river that was predictive of an in-river fate?
It was associated with poor performance in the river.
Okay. So, Mr. Lunn, if we could turn to the top part of Figure 1A of Exhibit 558. Sure. It's page 214, which will be the second page. How many fish are in the group with the mortality-related signature in Figure 5A?
You're not showing 5A.
Oh, sorry, 1A.
Oh, I'd have to read through the paper again. Okay, so over all of those fish, there's somewhere around 40 fish in A. Is it A what you're talking? I'd have to actually look at the paper to remember the exact numbers. Do you want me to look at the paper? Which tab is this?
This is...
It's outlined in the paper.
Yeah.
Which tab is this?
It's 18 in the commission's binder.
I can suggest to you that it was 10, but, you know, if you could...
Oh, I'm sorry, I misunderstood. I thought you meant in the whole study.
No,
You're
I'm just speaking about the fish with the mortality signature in Figure 1A.
Okay, where we've demarcated it. Yes, I believe there's 10.
Now, when I count the two groups, it looks like there's samples of 5 and 5, and I understand from speaking with others that that's actually a mistake, and it was 6 and 4.
Yes, there was. We've had a discussion about this with Gary Martyr. The top bar that demarcates survivors and upper river morts, there was a glitch in the way that was put on, and there's one fish that died that's missing on the left-hand side.
Okay. But certainly, for the purposes of your analysis, you didn't use 5 and 5, you used 6 and 4.
No,
so it's just a
I know.
glitch.
Gary Marty has had all of the data and redone all of the analyses, and we noticed that we had a glitch on the top bar.
Okay. So for this purpose of my next set of questions, it's going to be about determining whether or not a diagnostic test can be derived from this, because I'm anticipating my friends might recommend that we could try to test for these fish using the genomic signature. So that's going to kind of form where I'm going. So essentially, 40% of the time, you would be incorrect if you tried to predict based on this test?
40% of the time you would be incorrect. I think there's something that is in the science paper that I need to explain, and that is that what we found with this signature, and it makes obvious sense when you think about it, is that the individuals on the extremes of this signature, the individuals that are most highly affected, which are those on this figure that would be way to the left-hand side, as you go on this almost continuous gradient, you have individuals that are highly affected, individuals that are something in the middle that don't really have any effect, and then you have individuals on the other side. If you envision this as individuals having exposure to a flu bug and having the flu and having pneumonia, right? So you have a very different probability of survival if you have pneumonia than if you simply were exposed to the flu bug. And this could be, it doesn't even have to be a disease scenario. If you had individuals that were starving and some that were nutritionally deprived and some that were well fed, you would only expect that there would be an effect on survivorship on the ones that are starving, not the ones that are just nutritionally deprived. And so this is the way that we've looked at this data, and others have as well, and in other kinds of studies. What we're looking for are the physiological extremes. And so you're absolutely right. There is no predictive power for fish that are intermediate in this signature.
Now, Dr. Garver, you have experience in developing diagnostic tests for developing viruses?
That's correct.
Okay. Would you use or recommend a diagnostic test from this data?
To determine a virus,
Correct.
a specific virus from a genomic signature?
That's right.
I think if you had a biomarker for that virus and you had validated it in a lab, then yes, you could use genomics to identify that virus.
Okay. Now, in this case where the ocean tagging study is only predictive 60% of the time, is this data that you would find suitable to create a diagnostic test from?
Well, like I said, you have to identify that signature to ensure that it is definitively just to that virus. For a diagnostic assay, you have to ensure specificity and sensitivity. So in other words, you want to ensure that you have no false positives. And to do that, you need a specific biomarker for that virus. So if you're able to identify a specific signature for virus A, then yes, you could use a genomic profile to diagnose that. But typically, if you know what the agent is, you're going to seek the agent. You're going to look for the agent. You're not going to use genomics as a diagnostic. You potentially could, but why would you? If you know what virus you're looking for, you're going to look for the virus.
Okay.
And I'd like to add to that, and I think I've iterated this a couple times. The genomics approach that we use is for discovery, and that's its sole purpose. It wasn't that we were going to run microarrays on every fish and predict their survivability. The point was we don't understand, or we didn't understand, what was undermining performance in the river. We used genomics to try to understand more about potential mechanisms that may be involved. And in this paper, it's very clear that we're not expecting to find a single physiological component that can predict all mortality. It would be completely unrealistic. Fish are hit with all kinds of other things in the river. The important point of this work was that for a segment of the population that was coming back into the river, they were so ill affected that there was an effect, even before they hit the river, on their subsequent survival.
Now, the genomic signature affects different sockeye stocks differently in freshwater, I understand?
What we found was that in the freshwater one, we actually had we had a larger sample size. We had more fish that were tagged, so we were able to derive a study that contrasted 3 different stocks. And the reason we were interested in that was that there's a lot of work that shows, that stocks are differentially affected by different kinds of physiological components, disease being one of them, but we didn't actually set out to do disease here. We were interested in, if we found something that was predictive of survivorship or premature mortality, how predictive was that across different stocks? What you need to understand about the 3 stocks that we looked at was that they have differences in their susceptibility to high water temperature stress. And so I think the backdrop to everything that we have here is that the signature alone, or even a virus alone, in certain environments may not have a negative impact. But when you put something that might compromise fish on top of stressful conditions in a river, like high water temperature stress, and my colleagues have done a lot of work on high water temperature stress, you have a greater potential of having a negative impact. And so the stocks that would be most susceptible to high water temperature stress were the Lower Adams, which is a late run stock that's been entering the river early, and the Scotch Creek. Chilco, and there's a publication out of Tony Farrell's lab, is a superfish. It can take high water temperature stress, and it can have no ill effects that are measurable. And so the stock that really showed the least proclivity to responding in a negative way to this signature was the Chilco fish, which didn't have the double whammy of carrying the signature and being stressed by high water temperature. And that was one of our hypotheses for why that stock was much less affected. I have others, but we don't need to go into them.
So to summarize, for the freshwater study, the survival of Scotch Creek fish correlated with the genomic signature, and Chilco, and I'm getting from your paper late Shuswap as well, didn't correlate?
In the freshwater environment, there was not a correlation between survivorship of those two stocks.
So you'd agree then that the freshwater tagging study is not consistent?
You know, what's interesting about the signature is that the signature in the marine environment is suggestive of a very early stage recognition of a pathogen. It's an immunosuppressive signature. The signature that we have in freshwater, although there are enough elements to find a good correlation between those signatures, is something of a change in that you move from a very early stage recognition signature to a full-blown apoptotic, which means cell death and stimulation of inflammatory response. And so it's more likely that it's at that point that if there is disease, that's when disease is starting to come about. And so one of the things that we hypothesize, and I think we might say something about that in this paper, is that at the point when fish are entering freshwater and swimming through freshwater, we hypothesize that it's possible that if there is a virus, that virus might be being transmitted at that point. So when we're picking up fish at that stage, you know, there's fish that are already affected, but there may be more fish affected as they migrate. And so if the point is that if we sample them as early as in the marine environment, if they already have that signature in the marine environment, they're more doomed. They would have had it for a longer period of time.
Have you done any follow-up studies on freshwater environments and similar to your science paper in subsequent years?
We're doing that right now in 2010, and we have a 300-fish study, the same 3 stocks, so we'll be able to look at this relationship again in those same 3 stocks. The difference is we tagged all of the fish in the marine environment. So, you know, for again, bringing this back to practicalities, one of the points in doing this was to provide something that might be useful to managers. Managers want to know if there is predictability on what the effects on salmon returns might be. They would like to know that before they hit the river. They would like to know that in the marine environment. This study was encouraging that we could identify a signature associated with poor performance in the river before they made it to the river. So that's before they opened the major fisheries in Johnson Strait and Juan de Fuca Strait. And so we decided this year to focus, or in 2010, to focus our efforts on the marine environment, but still looking at mortality in the freshwater environment. Because if we were to develop tools from this, that's the point where managers want that ability to predict.
And do you know when those studies will be published or be available?
We just ran the microarray study. We actually got some. The unfortunate thing is it's a new array. It's not the same array that we used. The array that we used here is no longer available. So we're using a different array which has a different complement of genes. But the results I've seen so far are very encouraging that we do have some important signatures associated with survivorship.
Now, I also understand you did a spawning ground study as well.
The spawning ground study was a bit of an add-on. There was a graduate student in Scott Hinch's group who was looking at factors associated with pre-spawning mortality at the spawning grounds. And so, yes, there was a tagging study, and we were able to get her fish and use them in our study as well. This came quite a bit later than the other studies, and this is what held up publishing this, because we wanted to wait to look at that third environment.
Okay. And I understand that 3.7 was the odds ratio for that study, and that as a result, they were not statistically significant.
It really was. We were looking to see whether or not it was consistent, but the sample sizes for that study were significantly smaller, and because that's all the fish we could get. So recognizing that we were really on the limits of being able to find something that was truly significant, the trend was still the same.
So then for the 3 studies, again, the genomic signature isn't predictive for a diagnostic test for predicting mortality. You'd agree with that?
I would say that the point, the first and foremost point of this study was to uncover potential mechanisms associated with poor performance in the river, and also have a better understanding of whether or not salmon were already physiologically compromised before they reached the river. I would say we have absolutely done that. I would say in terms of the predictive nature of this is a 1-year study, and clearly one needs to do this in more years and with more fish in order to really develop something that's truly predictive. So I wouldn't want to go out tomorrow and say, I have a test, to predict mortality in any year in any stock.
Have you done any analysis on whether or not the differences in gene expression are a result of chance or not a result of chance?
I don't know what kind of studies you're imagining, but randomizing samples, are you thinking technical, or I don't understand the question.
Oh, I'm trying to find out the confidence level.
That there is a signature?
Right.
Well, I mean, we've demonstrated this same signature in gill tissue in multiple years. We've demonstrated a highly correlated signature in other tissues that do not correlate with any kind of technical effects of running microarrays. Those when we can get correlations of over 0.95 between different studies for this signature. So I do think that we have validated that this is something biological as opposed to technical.
If we could turn to Figure 3 of Exhibit 558. As I see it, there's maybe if you could explain this figure for the commissioner.
Okay, this figure shows biological processes, and the biological processes are shown on the left. Provirus integration is a biological process. These kinds of biological processes are called gene ontologies, and for every gene, it is involved. Each gene is involved in a number of different biological processes, and so when we do an analysis, this is called a functional analysis. We're looking at all of the genes that are present on the array, and what are all of the biological processes, or GO terms is what as people call them, that they are involved in. And then we're looking, if we have a list of genes that defines a signature, what among those biological processes are statistically overrepresented in that list of genes. And so that's what this figure shows, are the biological processes that are found statistically associated with this particular signature. So on the bars on the graph, the ones that go to 0 to -3, those are processes that are downregulated, so that think of it as pathways that are being turned off in fish with the MRS signature, and the ones that go from 0 to 3 are pathways or go terms that are turned on in the fish with the signature relating to higher mortality.
Now, I understand you had some complex results, and in 7 of the 40 biological processes, some were at the same time down-regulated and upregulated.
That means you really can't describe, you really can't prescribe which direction the pathway goes. That is quite common in microarray studies, that sometimes it's very clear what your directional. What you have to understand about the technique is there are genes that can be negative effectors and genes that can be positive effectors of a pathway. So you can have a gene that actually, it can, when it's turned on, it's turning the pathway off, right? And then you can have other genes that when it's turned on, it's turning the pathway on. So it is quite complex. So when you get this, when you get this mixed pattern where it's not really clear that most of the genes are being stimulated, then you have to go and say, okay, what's the effector of each of these genes? What do they do? And is it that you have ones that are being turned off or actually the repressor? So it can be quite complicated. We didn't go into that detail here, and most people don't. Most people simply show which ones are obviously being activated and deactivated, and that's what we've done.
I understand for the purposes of your science paper, the samples were taken from the gills, and that's not the ideal sample, and specifically the ideal sample are heart, kidney, or brain, and I understand you've done that subsequently.
Yes, the reason we have to take samples from a gill when we're doing radio tracking is because it is a non-destructive tissue. You can't take the brain of a fish and have it swim to the spawning ground. So it really is our only choice. We can use gill, we can use skin, we can use muscle, but it's absolutely true that you are limited into what, if you're going to combine with radio tracking, you're limited in what kinds of physiological processes you can look for. And we have followed this up using destructive samples from other tissues.
Okay, so my question is, I understand that you were only getting inconsistent results on an individual level, where some would have brain tissue that showed the MRS and others would show heart tissue only that had the MRS.
I don't understand the term only, but yes, we do find, and this really was highlighted in our 2005 studies, where we looked at the same fish over 3 different tissues and showed that the signature, the highly correlated signature, was present in each of those, but the prevalence in each of those was really quite different. And the prevalence in that study, I believe, was highest in the brain and lowest in the liver.
And I'm assuming there will be more research on that in the future, that you'll narrow that down.
Absolutely. I mean, right now we're not doing a lot of work on adults other than the work that combines the radio tracking with the genomics, but we are doing a lot of work on smolt, so we will have a lot of information about we're running smolt studies using multiple tissues, where we'll be able to say at an individual level what proportion of individuals carry this signature in 1, 2, and 3 tissues. I should add that with the virus, the parvovirus that we've identified in tissue that contains this signature, kidney is actually probably the best tissue we could have been using.
Now I'm just moving on to the histology samples that Dr. Marty analyzed for yourself.
Yes.
I understand that his conclusion was that the lesions were related to blunt force trauma.
You're talking about the brains.
That's
so
right.
we sent, in 2009, when we first observed what we thought were abnormalities in the brain, we had histological slides made of those. Those were performed, the slides were made in our fish health group. They took 2 slices from the middle of the brain and made slides into them, or made them into slides. They took 12 brains total. All of those brains came from the spawning grounds. One of the issues is that when we're doing our genomics, as I said before, we're doing the genomics and we use the whole brain. So, and the other thing is that when we're doing the dissections or even the collections, we don't collect histology grade brain. We collect these samples for doing RNA work, and the kinds of chemicals one uses for that are different from the kinds of chemicals you'd use for histology. So the only reason I'm bringing that up is that those were the only brains that we had that were collected that were either collected in a chemical and never frozen, which you can't freeze if you're going to do histology, or they were collected in histology chemicals. So although we'd seen these what we thought was anomalous in smolts and in other points along the migration, the only samples we had available to run histology on were spawning ground samples. And so Dr. Marty got 12 brains that we had classified according to whether or not they contained these, what we thought, again, were lesions. When he read those slides, he concluded that they were likely arising from hemorrhages, and they were likely arising from hemorrhages as a sampling artifact, as an artifact of the handling of the fish.
I just want to make clear, I mean no criticism by that question at all.
I'm just being clear. I'm just trying to be clear.
Exactly, and I just wanted to clarify that point so we could nail down the brain tumor issue.
That's fine, yeah.
Now, at this point, you're not in a position to definitively identify the cause of the MRS as a novel virus.
The cause of the MRS yes, we have not definitively established that the parvovirus causes the MRS. That is something that is the topic for our disease challenge work.
And at this point, parvovirus in fish, this will be the first time it's ever been identified, if it is ultimately identified by yourself.
That is correct. This is the first time a parvovirus has been identified in a fish. They have been identified increasingly in lower vertebrates in the last decade, generally associated with mortality events.
Okay. And other parvoviruses have been identified in humans, dogs, and sea lions.
Oh, and shrimp and insects and ducks and geese and a variety of other lower vertebrates as well, and snakes.
And Dr. Garver, what are your thoughts on whether parvovirus is linked with the MRS?
I agree with what Kristi says. At this time, we don't have the actual link to the fact that it is the cause of the MRS, and that's what we're working towards.
Now, I understand that you have identified a 2,214 base pairing sequence.
That's correct.
And that's about 50% of the parvovirus
Genome.
Yeah.
Yes.
Does a partial signature necessarily mean the full sequence is there, or is it a possibility that it could be chance?
I guess I don't see how it can be chance given the kinds of data that we're seeing. Chances of exactly what? It's certainly not endogenous in the salmon genome. It's not something that is there in the DNA of the salmon. So I don't know where you'd pick up a partial viral sequence by chance.
And at this point, have you done any histopathology to determine if the genomic signature or parvovirus is associated with disease?
That, again, is something that we're going to concentrate on with the disease challenge work. We have done a little bit of histology, taking some fish that were parvovirus positive that were sampled, some smolts sampled in the marine environment. The thing to recognize is that when we sample fish in the marine environment, at the time that we're sampling them, we're sampling live fish. We're not sampling fish at the time of death. And I am not a histologist, but from what I understand of histology, the histology will become a lot stronger and more powerful and easier to detect if you're sampling fish at the latest state of disease. And a lot of histology that's done in concert with disease, but not all. Gary Marty has a study on herring that looked at wild herring, has concentrated on moribund fish or fish that are sampled close to death. So when he processed these, we only looked at about 10 or 12 samples, and he didn't see anything that through histopathology was really conclusively suggested that there was a histological feature that would be associated with mortality.
Okay. Now, while I understand you're getting closer to identifying parvovirus as the cause and studies are ongoing, there still are alternative hypotheses or differentials that it could be related to?
I just rise because the way the question's framed doesn't seem to accord with the evidence. She says, getting closer to finding that X is the cause, as I heard you.
Well, what I meant by the question was studies are starting to, there's different possibilities still. They're looking at one hypothesis, but at the other time considering alternative hypothesis.
I would say that's correct. I mean, at the moment, we have enough evidence to keep going with the parvovirus and keep going with the research to establish whether or not the parvovirus is A, associated with the signature, B, causative of the signature. If we find that it is not, then obviously we have a couple things to do. One of which is to go back and say, well, okay, what else might be there that we're missing that might be associated with the signature? And maybe there's, you know, another infective agent that we have missed. But I think we will still continue on with our work on this parvovirus because we've identified a virus in sockeye salmon that is at a very high prevalence, and at a very high prevalence at the time point when sockeye salmon are entering the marine environment and undergoing some of the highest mortality that they do in their life history. And so I think that in and of itself warrants further study into this virus, even if in the end it doesn't happen to correlate with the MRS.
Okay. And Dr. Garver, do you have anything to add?
No, just to the fact that we're following the scientific methodology and the approach. You take one step at a time. You need to confirm route A before you can go to B a lot of times. So that's the project that's the track that we're on right now.
Now, I understand in your timeline document, and I'm specifically referring to Provincial Tab 20, which is the earlier one from May, as opposed to the later one, that you identified a declining prevalence of the signature in the ocean, but could point to a potential impact of the signature, or, you know, and then goes on a little bit to, or early marine mortality, and then goes on, but cannot discount the possibility that some individuals recovered from the signature?
That's absolutely correct. What we're doing at this point, in addition to doing the challenge work, is to starting to accumulate the information about how prevalent is this, and do we see shifts in prevalence over space and time, or in years where we have strong year class strength and weak year class strength. When you're working with wild fish, looking at shifts in prevalence is one of the indicators that people use to try to pinpoint what factors might be involved in declines. I should point out that there's studies on disease that have taken place in Oregon on wild Chinook and coho salmon from the Columbia system. And one of the things that they found, they look at BKD and they look at various parasites. And I believe Mike Kent was even involved in some of these studies. It's out of Kim Jacobson's lab. And what they have found over 10 years, so they've been at this longer than we have, is that, and they only sample fish in June in the ocean. What they have found over 10 years is that they can't, in years where ocean conditions are poor, they can't find a fish with BKD in the ocean when they go out and sample in June. Now, recognizing that when they go out and sample in June, the fish have been in the ocean for a month and a half or 2 months. If they go out into the ocean when the conditions are good and good for growth of smolts, they can find up to 70% of the smolts that contain very mild infections with BKD. And they found a very similar result when it came to parasites. They can't find fish that have a heavy parasite load when the ocean conditions are poor. They can find fish with 3 or more parasites when the ocean conditions are good. And what they have concluded is that fish disease is not tolerated when the ocean conditions are poor. Those fish that carry disease simply don't survive. What they've missed in their studies, however, is that they don't link it to the freshwater. So they don't really know on an annual basis how many diseased fish might have gone out into that environment. And I would say, that's something that we're interested in as well. So right now, with the declining in prevalence, we're looking for patterns. And the signature in 2008 and in 2007, mind you, small sample size, showed a declining pattern in the prevalence of the signature. We have since now also focused on this parvovirus, and we see the same thing. When we see the parvovirus, the highest prevalence is in the river. We see a declining prevalence in the early ocean environment, but hints to that decline may vary in different years. So the degree of decline that we observe may not be the same in every year, and that is what I'm focused on, is how much does it decline. But you're absolutely right in that we do need to establish whether or not fish could recover, and or whether a decline is always going to be associated with mortality, and we have not established that. The other thing that I would just like to say is that with the parvovirus, we have been sampling a broad range of stocks, and we've sampled a broad range of stocks in the Fraser River. In last year, in 2008, there were over 400 fish collected in the marine environment in May and June in the ocean, and we looked at the presence of this parvovirus sequence in those fish, and the only stock that we could not find the parvovirus in, that we had a sample size of over 15 fish for, was Harrison, and we had 51 Harrison fish. Harrison is the one stock in the Fraser River that is increasing in productivity. Why that's important is that we're looking for patterns. Okay, we don't have all of the data to show disease and to show mortality yet. We're working towards this. We are working with wild organisms. But everywhere we turn, we see indications that indicate that what we're looking at could have a negative impact at this life history stage.
Thank you. Those are all my questions.